Hello.
Hello, yeah, we are all welcome to this session. My name is Dr. Muhammad Musa Lawan, the Deputy Vice Chancellor, Academic Services of Yobe State University. Today, we'll have, at this session, we're going to have Professor Amadi Ihunwo, who is going to talk to us on undertaking neuroscience research and managing collaborations. But before I give Professor Ihunwo the floor to give us the talk, let me quickly read his bio. Professor Amadi Ihunwo is a professor of anatomy and neuroscientist in the School of Anatomic Science, Faculty of Health Sciences, University of Witwatersrand, Johannesburg, South Africa. He leads the comparative adult neurogenesis. Professor Ihunwo holds a bachelor degree in medical sciences from University of Port Harcourt in anatomy in 1984, Masters of Science from University of Calabar in 1992, and a Doctor of Philosophy from Mbarara University of Science and Technology, Uganda, in 2004, with research conducted at the Vollflechsig Institute of Brain Research, University of Leipzig, Germany. His teaching, research, and administrative experiences in the university spans over 35 years. That is from January 1986 to date. 11 years in Nigeria, UNIJOS specifically, and Bayero University, Kano, 8 years in Uganda, and 16 years at WITS. He had proceeded to Uganda in 1997 under the Technical Aid Corps, a program of the federal government of Nigeria, and completed same in 1999. In 2000, Mbarara University requested his return to assist in reorganizing the anatomy department that led him to spending another 6 years and becoming the associate dean of the School of Medical Sciences. Professor Ihunwo joined Wits University in 2005 specifically. He completed a 5-year research project as the principal investigator under the Switzerland-South African Joint Research Program, followed by a South African National Research Foundation, NRF, research project. He is a C-rated researcher under the NRF. Professor Ihunwo has graduated MSc and PhD students and has several publications in internationally recognized journals. In 2016, he was a recipient of the WITS Faculty of Health Sciences Most Prestigious Award. That is the Exceptional Service Award. And in 2017 and 18, faculty recognition for dedication and achievement in research. In same 2017, he received the Distinguished Alumnus Award of Excellence from the University of Port Harcourt, Nigeria. The academic citizenship of Professor Ihunwo extends several universities in Nigeria and Africa. Professor Ihunwo is currently the Secretary General of the Society of Neuroscientists of Africa, SONA, and member of the Governing Council of the International Brain Research Organization. He was recently re-elected for a second tenure as the counselor representing Africa on the Council of the International Society of Neurochemistry. Through this, several Nigerian and African postgraduate students and staff have received funding to travel and participate in international neuroscience schools, workshops, and research visits to other laboratories. He is a visiting scientist at the Brain and Mind Institute, Aga Khan University, Nairobi, Kenya, from where he is joining us now. Professor Ihunwo, you are most welcome. You have the floor now. The floor is yours. Thank you.
Thank you, Mr. Vice Chancellor. I just want to confirm that I am being heard.
Yeah, you are.
Okay, thank you, sir. And I'm very, very excited on this occasion. And my excitement comes from being privileged to have had a chat with Mahmoud just about the time he was rounding up his PhD, and he asked me, Prof, what should I do? And I remember Mahmoud telling you, I don't want you to get lost in transit. I want you to go back to your university, let them know you have finished your PhD, and tell them what is opening up for you. Mahmoud, I'm very glad that you heeded that. And today we're talking about a laboratory at Yobe State University that you and your group have put together. It's quite interesting that I was given this session, the last session. I've followed up the presentations, and I find that the only way I can contribute to this is first to share my experience, as a kind of researcher that will come to Bio Research and Training Center. And I know a lot of our colleagues in Nigeria will find themselves in my story and desire to come to that lab. Then I will just share briefly on some things we've done in comparative adult neurogenesis. Then I will give my answer. In the course of the presentation, I think I'll be making some references to what I envisage RTC, BioRTC will be managing in this time. Now, just like any researcher starting out my first degree, I had to do cephalometry, measure the head, and subsequently talk about the shape and relate that to what is the likely cranial capacity an individual may have. We talk about in terms of dividing it into three parts, either brachycephalic or mesocephalic or dolichocephalic. But of course, knowing that that is just a cephalometric research. Then I proceeded to my master's at University of Calabar, where I started real laboratory exposure. Was looking at streptomycin on fetal brain in the Wister rat. And of course, in the course of that training, you learn techniques, research techniques, laboratory techniques, which you are not privileged to undertake yourself. You're not privileged to have a hands-on. However, those knowledge were kept. And by the time I had the opportunity to go for my PhD and do a laboratory research, I moved on to use an animal model, the TG2576 mouse model. And that gave me an opportunity in Germany to be exposed to immunohistochemistry, to stereotactic manipulation, to electron microscopy. But before I went to Leipzig, after my master's, I went to University of Maiduguri. As at then, University of Maiduguri had an electron microscope. When I traveled to Maiduguri, yes, I could see the machine, but it wasn't working. So that desire is something I see that the BioRTC, you will become a site where people will be coming, and you must prepare yourself as a training center for research techniques. I expect you to prepare yourself in that area, that it's not just coming in to do research or complete a research protocol, but I see people coming to BioRTC just to learn a technique because of the proximity of your location in Nigeria. So that is one of the things you'll find that will be happening to you as a training center and a research center. Now, if I speak briefly with respect to my area of research, we came into an area of research that the dogma was there that we don't have cell proliferation in the adult brain. And by the time we came in, we had to generate questions and concepts, and I see you doing that in BioRTC. You have to generate questions. I mean, these are kind of the questions we're asking ourselves in comparative adult neurogenesis. Say, is that pattern of adult neurogenesis in laboratory animals the same in the wild? Now, for some of us, the word wild has to follow. We prefer to speak about natural population. I mean, the animals are in natural population. The animals we had to engage with are animals in natural population. So when we seek to the world wide, it look as if something scary that you cannot go and engage, but that is not true. So if you understand the environment and understand that this is the natural environment of an animal, then we're looking at animals in natural population. We asked ourselves the question, is the pattern of adult neurogenesis in mammals consistent through phylogeny? Because there are two sites, an active site or a potential site where you can have cells being generated in the adult brain. Then we asked ourselves, will the pattern be consistent in animals within the same order from natural populations? And I will just share with us briefly a kind of diversity in terms of the order and the family of animals we've had to engage with. Then what is the influence of the environment? We look at the environment, you look at seasonal changes, start asking yourself questions. Could this influence adult neurogenesis process in the natural environment? We understand there's a genetic part. We also understand that the effects in disease state. So these are kind of questions. What is considered an adult animal in the natural population? So we have to define that. And what are the age-related differences? So one thing you'll find with a lab, just like yours, is that you must continuously generate diverse research questions. That is one feature that you must have at the BioRTC. Keep on generating research questions. Keep on looking at concepts that can be changed because you've set up this environment. Then we personally, I mean, when I started engaging comparative adult neurogenesis, the first thing I did was a review paper with one of my students. And if you check that word, adult neurogenesis, as a search phrase on PubMed, as of today that I'm speaking, there are 11,963 publications in circulation in PubMed. If you engage other data systems, you will have more than this. But when we did this paper on neurogenic sites in adult mammalian central nervous system, only 2,386 publications were there. So that is how I found myself in this domain. And I said, as at that time, is there any animal within our South African environment within that literature? There was none. And I had to take advantage of a colleague of mine, Professor Paul Manga, who was working on evolutionary neuroscience and who was interested in African animals. And I said, look, I think I need to look at this process, you know, in species that are on our African continent. Now, just to understand that concept of adult neurogenesis, what happens is that we have cell proliferation in the dentate gyrus. Dentate gyrus is part of the hippocampal formation. So it starts off as a stem cell, then immature cell, then immature neuron, then mature neuron, then it becomes integrated into the system. Then another site is the subventricular zone, where we have migration of cells going to the olfactory bulb. So these two sites are considered as the active neurogenic sites. And what happens is that from that germinal layer, the stem cell niche is generated and they become integrated and functional neurons. This happens in the adult brain. In addition to that, we also have what we call potential or non-neurogenic regions, where again, there's a stem progenitor cell, which will give rise to an immature cell, and another immature neuron, mature neuron, and some of those functions are still questionable, and these can be influenced depending on the environmental stimulus or the disease state in that situation. Now, of course, with time, we're having markers that are able to, you know, identify what phase or stage of adult neurogenesis that's taking place, whether it's a cell proliferation stage or cell genesis itself, or the specification when there is differentiation, and when you have a full neurogenesis and functional. From our side, we have also started looking at the gliogenesis component in terms of cell proliferation in the brain. So we started off simply using, again, a rodent. This is the four-striped mouse, which is found in Africa. And we were able to observe that this is a subventricular zone. You have the generation of those neurons. This is the rostral migratory stream. So you could see, I mean, using the Ki-67 that yes, there is cell proliferation taking place, and there is a rostral migratory stream, which is the migratory process involved in adult neurogenesis. In the dentate gyrus, we could see in the subgranular zone these cells that are generated, I mean, within that animal species. So we moved on again, with animal that is very common around us, the common mole rat. We could also observe the same process taking place in the lateral ventricle and migratory process, and of course in the olfactory bulb of the common mole rat, you can see distinct cell proliferation. In the dentate gyrus, we could also see this neuronal cell proliferation that is taking place, and using the DCX, we could also identify the mature neurons in the that the idea that yes, we could have potential neurogenic sites. I mean, the cortex layers 2 and 3, we could see cell proliferation taking place, and also somatosensory cortex. So we were not just now limited to the active sites. We could understand that this could happen in other areas. Now, from our side, we've taken that further, looked at several animals, and these people just spoke to a number of animals that we had looked at this process. And this is a kind of breakdown of the different order and the families of the animals that we've looked at different times. And so the Eutheria carnivora, cetacean, Arctodactylia, rodentia, micro and megabats, primates, and just did a diversity of that. And it's quite interesting that just doing that, one could establish that from an empirical data point of view, that this process takes place in this dentate gyrus, specifically in this publication. So we have an array of animals, the arctodactyls, the afrotherians, examples, the rodents and bats, that this process takes place. Even in the lion, the African lion, we could again establish the same process, the dentate gyrus, that we have the processes taking place. The river hippopotamus, West Indian manatee, herb cells give us positive, stating that yes, the process takes place. However, in the Habopopis and minke whale, which are cetaceans, we did not notice any of this cell proliferation in the dentate gyrus. Now, one would wonder what is going on. Of course, my colleague had proposed in terms of the thermal regulations associated with this animal may be the cause of that. But when we look at the piriform cortex, we could identify that yes, that this process still takes place in the. Now, again, with this way we generated question, part of the question that was coming was, how do you distinguish an adult animal from the natural population? Now, in the literature, you find some of them, you can tell which is under the sub-adult or an adult animal. We started out off using a South African ground squirrel. One of my master students, Samson, looked at those sub-adult and adult. So we started introducing the staging of the animals. In the sub-adult group, you could see that, yes, the process compared to the adult is much more. This is cell proliferation in rostral migratory stream in a sub-adult and the adult group. Then you look at the density of the young and immature neuron along the migratory stream. This is a sub-adult and this is an adult. So we can comfortably say that with age, which is normal phrase you hear, that we age, there is a decrease in adult neurogenesis. The same way, again, we interrogated the dentate gyrus in terms of the proliferation of cells, subgranular zone in the sub-adult compared to the adult. In the same way, immature neurons using the DCX in the sub-adult to the adult. So it's very clear that one could speak to the fact that, yes, we have that process in a declined format. I mean, from as the age progresses. I mean, did a quick count, and could see the difference between the sub-adult and the adult number of neurons, Ki positive neurons in the dental gyrus. Now we've moved on and we've done several other animals, but we decided to also look at the avian species and see what the scenario is. And we could first tell that there is a difference between the two. However, we could also specify at which point, and that is why we started with Japanese quail. We said, can we tell the point where this process will peak in an animal species, in an animal model? And for the Japanese quail, we could establish that at 5 to 6 weeks. I can tell you, if you want to use the Japanese quail for any experiment and you're interested in the process and to see how it's been affected, you should use the 5 to 6 weeks of the Japanese quail. And that is what we found. And we're using that as a basis time subsequent work on the Japanese quail to consider whether we're looking at what will affect the process and how the process is affected. And that's what one expects that will be happening with the animal investigations. Of course, we've looked at over 7 other avian species that are from the natural population. And we are establishing the same thing in that process. Now, having said that, and from all that has transpired this past 2 weeks, for me, my counsel to the BioRTC is that comparative animal model is a must for you. I mean, we've spoken, there have been presentation on different models, and I'm aware that Mahmoud and your team, you've been involved in trends and you've looked at different animal models. BioRTC has to maintain this approach of having a comparative animal model in the investigations that would take place there. My other counsel is that you be open and have, I mean, again, within our Nigerian environment, we have animals that are unique in that environment. We have animals that are also becoming endemic. And I would like to encourage you to partner with veterinary anatomy departments and explore that and establish and provide us with data, empirical data on these species that are there. James Olopade has alluded to the fact that environmental neurotoxicity, I mean, in his general terms, the environment has an influence that we must consider, and using animals within that environment where humans are is an avenue to extrapolate what could happen. I like the last presentation emphasizing that need, that we need to, again, not just wait until we have women. So we can use the animals in our environment to speak to situations, and the environmental influence on the animals is also possibility of that happening in the humans there will be there. So much fundamental research is still needed, and a lab that the BioRTC is very much needed at this point in time. Then creating animal models for neuroscience research. I believe BioRTC, you can do that. You can use that and create for yourself an avenue. Even the health disease models can start looking at that possibility. I don't want to believe it's only when you have a transgenic animal that you can do an acceptable animal, create an acceptable animal model for research. You should look into that. And as you go on, try to start creating animal models that will be part and associated with the BioRTC. Now, I would like to go to funding, and I'm glad Professor Sen yesterday made reference to that when you asked for his advice. And I want to appeal to the vice chancellor, through you to the vice chancellor, and also to your state government. This is a special lab. This should be encouraged. Financing in our setup, you know, we need to, when we identify something special, we should also give it a special attention. And I would like to plead for that dedicated funding that could be in the budget, you know, for this laboratory. And of course, with time, we could also be talking about counterpart funding, you know, in such an environment. It will be very helpful to the BioRTC to work with that. I also want to subscribe totally to what Mahmoud has said in terms of facility charge. I mean, that should be planned out and made part of it. You can run training for techniques, not necessarily when I want to do a project, but just the techniques. based on the equipment that are coming in into that laboratory. You can just run a training, create training programs, workshops, just knowing how to use those machines to tie to your funding profile. And finally, because of the second half I wear in terms of managing societies, I just want to refer to the fact that for BioRTC, I have envisaged a kind of managing collaboration and design that will be bringing professionals and institutions and sharing resources. And this seems to be specific to science. And I always like to propose the Drucker's design. And you must remember that people will collaborate when the benefit outweighs the effort. And the purpose of BioRTC should dictate the fit of the collaboration network. So you have to take on that role to define the areas of collaboration that will be beneficial to you. And every network has a choreographer, chief organizer, somebody who drives it. Mahmoud, we've seen you at that and your team. And define your governance structure and behaviors to manage collaboration. So that is very helpful that as you set up this lab, as this lab goes into motion, we must, again, it's very clear your team, through trend, you've been involved in innovation, in designing laboratory, small laboratory equipment and dealing with techniques which you have displayed at workshops under sonar during sonar meetings. And I still want to encourage that. I'm very confident that people can do that in this environment, in this lab that is being set up. And from the society point of view, I mean, SONA, the ISN, the International Brain Research Organization, we want to see the BioRTC engage with these societies. And that means you must be ready to host researchers on short visits. It could be an established researcher, it could be an emerging researcher, it could be a postgraduate student. So you are aware that, funding are provided to these different categories of people to undertake research visits. So you must prepare your environment as you prepare the lab, prepare the environment to receive people accommodation-wise, prepare environment for the mobility of people, and be a training avenue, like I said. And your measurable output must be directed at research projects. It could be linked to an MSc project, it could be linked to a thesis, it could be linked to publications. So your training responsibility, which will attract people to partner with you, must be in this direction. And interaction with institutions and societies willing to recommend BioRTC for research purposes will create a visibility for BioRTC. Encourage application for research fellowship, research grants. And I want to assure you from our side, from myself, from the societies that I engage with, we will recommend BioRTC as a site for funded research visits. That you can be sure of. That I can guarantee you people that will happen, that BioRTC becomes a site for research visits to keep the activities going on in that place. So thank you for giving me this opportunity to just wrap up this session that you have speakers. And I want to say congratulations to you. And I can assure you I will make our time somehow to visit BioRTC. Thank you very much.
Thank you so much, Professor Amadi.